which you are also understanding. Now, the FASTA file, if you have to save the FASTA file, you have to save in the dot FASTA format, dot FASTA, dot FA, dot FNA, and dot FAA. In this file extensions, you have to save the FASTA. FNA is for nucleic acid and FAA is for amino acid, right? And this file can be opened in any text editor file like Notepad. Moving again. It's not moving. Just wait a minute. Just a minute. Okay. Anisha, pointer is not coming. So if I am using the pointer in the, I already told in the Zoom, file is not moving forward. So if you know how to use a pointer in the PowerPoint, then you can tell me. Else I have to go with this only. Pointer is not coming.
I don't think that that will be possible. So you can just use the cursor only. Can you try moving
Can you
the
see my mouse?
Yeah,
Can you see
yeah,
my
yeah,
mouse?
yeah. I can
Okay,
see the
that is
cursor.
visible. Okay, then fine, That is more than enough. Okay, fine. So guys, after this is the FASTA format I told you. This is the amino acid FASTA format, where there are the amino codes. There are the letter of amino acids here. Uh, so this is the amino FASTA file. Then now, after understanding only this is the only basic thing which you have to understand, the header and the sequence. This is the amino acid file. And now you have to understand just like a bioinformatician, and you have to answer the question I'm going to present to you. So the first question is it possible to detect the number of sequences in a FASTA file without manually opening the file? So can anyone answer this? Write it in the chat box. Is it possible to detect the number of sequences in a FASTA file without manually opening the file? Yes, Dimple is correct. Rashi, yes. Grep and WC minus L. Yeah. So if so, how? Yeah, very nice. So yes, by using command line, you can do this. Now the next question, very well. People are doing very well. So the next question is, if you scan a multi-FASTA file and find exactly 20 lines starting with greater than symbol, how many individual FASTA records are present? Answer. So there is a multi-FASTA file and you know that there are 20 greater than signs. So how many individual FASTA records are present in a multi-FASTA file? Correct. People are understanding. This is nice. So there are 20, if you are finding 20 greater than sign, it means there are 20 FASTA records in a multi-FASTA file. Now the next question. Okay, so which specific command is used to find and count the greater than symbols across the entire file? That you have already answered. Um, Even again, you can answer it. So this is the command actually. Yes, yes, guys have already given the answer before, so I showed it to you. Okay, using grep. So there are two ways of doing this. Actually, I use the second one format, but this is also a very good syntax where you use grep minus c. Minus c is okay. Just can anyone make me understand what is how this command is working? Can anyone mute yourself and explain me how grep minus c is working? No, c is not a character.
Ma'am, it counts all the samples present and give it the output, like all together, all the sample that is present in the file.
Yeah. So, Soumya, you are correct. Um, Actually, here you are minus c is for count. She is correct. Minus c is for counting. That's why you are using grep minus c, and then you are using a caret sign at the start of the greater than sign. Though there is no need of the greater than caret here also, because in the FASTA file, except the header, there is no greater than sign at any line. So without even using caret, you can work with this. So now, and the second command is grep greater than file name dot fasta, and then after using pipe, you can do the wc minus l. Wc is word count. So after that also, you can you can use the second command, right? Now the fourth question: if a file named file.fa contains 50 lines of text in total, can you determine the exact number of FASTA records? Why or why not? Anumita is correct around the patterns. Right. So for the fourth question, can you answer me? For the fourth question, can you answer me? Or if you have any doubt with any of the three, you can ask. So in the fourth question, if a file named file.fasta, there is a file with the name file.fa, contain 50 lines of text in total, can you determine the exact number of FASTA records? How? Can anyone answer and unmute yourself and answer it? Please unmute. Any one of you unmute Arijit. Can you unmute yourself and explain how can we answer it?
Yeah, so without knowing how many gated than symbols are occurring, we cannot really determine the number of faster records.
You cannot
Right.
understand how many, correct. You cannot understand with the 50 lines how many FASTA records are there in this multi-FASTA file.
We need
Because, yeah, speak. Um, So from the 50 lines, you cannot understand how many FASTA records are there in the file because only the header is fixed, right? Number of lines for the sequences are not fixed. There is a possibility that there is a FASTA file of only one FASTA record of 50 lines, right? So it's not fixed. So you have understood how FASTA is working and how you have to use FASTA sequences in the bioinformatics. Though you are going to learn a lot of things, but you are actually using your brain how you can figure out how many sequences are there, what is the formatting and everything. Now comes to the FASTQ. So FASTQ, you also know about it, that it's a FASTA file which has included quality score, which also have the quality score. Then okay. So in the FASTA file, this particular FASTQ format is widely used through all the sequencing machines like Illumina or Nanopore. History is it is developed by Wellcome Trust Sanger Institute in the late 1990s. File extensions, particularly for FASTQ, which are you going to use, you have to save your file or you are going to use the file in this .fq file.f file.fq or file.fastq, right? So you have to save this in this format, .fq or .fastq. Next is FASTQ is exclusively for nucleotide. It's not for protein. It's only for nucleotide. And also one more information that FASTQ is a very bulky file. It cannot be moved from on the internet as .fq or .fastq because it's very bulky. So we have there is a SRA file format which is a compressed version of FASTQ. This is present on NCBI, and you are going to use this SRA file from NCBI. These things will be given to you in information by in the next coming sessions. You will understand what is SRA and how you are going to download it and converting that SRA into FASTQ. Next, the format of the FASTQ. Now just understand the FASTQ format properly. So here you can see there is a header which is starting with the @ sign. Then there is a sequence, then there is a separator, and there is a quality of for each and every nucleotide, right? In the header, you have after @ there can be ID of the sequence, or there is the complete information of the sequencing machine where you have put your sample in the flow cell, what is the index number, or what is the indexing. Everything you will come to know from this information. So understanding the formatting, here this is the header which starts with @ sequencing, +, and quality score. Now what is the quality score here? Let me explain you here. There's a Fred score for this. This quality score is calculated through Fred score. The formula is this. And in this case, for example, when what happens actually during the sequencing process, when the sequencing machine is working, it calls the bases to make the read sequences. While when it calls the bases, there can be error in the machines, right? There can be error in the machine. So you can see that there is a possibility that one error, there is a stretch of 10 base calls, and in the 10 base call there is a so in a 10 base calls, there is a chance that there will be a one base call error. So if there is one base call error in a stretch of 10 base calls, it means there is a 10% chances of error. There is 90% accuracy and 10% chances of one error. So it is written as Q10 means 10% error. Q20 means there is 20% error. Q30 means 30% error. So to understand this, there is 1 in 10 chances of error. It's not 1 in 10 chance. This is not 1 in 20 chances of error. It is 1 in 100 chances of error. So it is 99% accurate. And Q30 is 1 in 1,000 chances of error. So it is 99.9% accurate. So you have to understand what is the Q value for each and every nucleotide, which is called in the sequencing machine. Once the sequencing machine get the Q score, it converts the Q score into a SI code, and each SI code is corresponding to a particular symbol, right? So for a particular Q score, you have a particular symbol. So now if you have a Q score 0, it means this you will get a symbol exclamation. You will get an exclamation sign. Exclamation sign means that there is a very poor quality base calling has occurred. Then in the Fred's looking to this, if you see there is HNI, they have a Q score of 39 and 40, and they are of high confidence, high quality symbols. They are the high quality symbols which should be there in the read sequences. So in the read sequences, FASTQ is showing you the quality scores by using all these symbols and showing you that you have to take the read forward or you have to remove all those reads. Normally, there is a 30 Q30 Q score 30 is used. Not as it's a standard. It's a gold standard in NGS that you have to use a 30. That you have to use a 30 score, and where you have in the 30 score Q30 means there is a question mark. So after question mark, all the nuclear symbols will be used in the FASTQ, but before the symbols below that 30 will not will we like to trim it, we used to trim it from the data. So you have to use only this symbols in the FASTQ files. Now if you have understood this and the previous slide the format, now you have to again think like a bioinformatician and you have to answer this. Is this is a read file, there are multiple reads in this sequence, and can you tell me is this sequence is a high quality or it's a low quality?
High quality.
High quality, yeah. How you come to know? Should I show you the previous slide? Yes, because of the I. So here I have shown that the I has the highest scoring and exclamation has the lowest scoring. So it means you can accept these reads because it has a high scoring, right? In this way, you are going to filter the reads which are of good quality and you are also removing the reads which are of bad quality. And you are going to understand in the next sessions properly how you are going to use it, and also you are going to you have the hands on over it. Now the next question is, you have a file named r1.fq. It contains millions of individual read sequences. Without opening the file, how can you determine exactly how many reads are inside? Like FASTA, as I asked, the same thing I'm asking for the FASTQ. Without opening the file, how can you determine exactly how many reads are inside? This is tricky. This all depends how you have understood these 2 points.
Ma'am, we can change the greater than sign with the at the rate sign.
The point is you are somewhere correct, but it's not properly, because if I tell you there is in this particular image there we have given a quality scores and at the rate is also present in the quality scores. So in the file there is a possibility that you have at the rate in the quality. So this is a tricky here. This is actually a trick here. If you even use the caret at the rate means at the start of the header there should be at the rate. That can work, that can may not be work, because quality score can be also be present at the starting of the uh, at the line, right? So no, the awk is not the answer. Can you think any actually bioinformatics is all about logic. If you find the logic, you have to see the complete file, you have to find the logic. Once you get the logic, you are done with your work. It's just like a game. It's like not like mugging everything. I told you 2 things here, that there is a header which have 4 lines. I'm giving you a hint that there is a format where there are 4 lines. Can you think of it? Because FASTA has multiple sequence lines, but FASTQ have only 4 lines. Like ID, you can use the separator. If you want to use a separator, also there is a problem with the separator. Pankaj is right. You are thinking well. I'm happy that you are thinking. But Shivangi also said separator, that you are thinking that there can be a plus sign, but plus sign is also here in the symbol. So again, you won't be able to understand how many FASTQ reads are there in the file. So this is are everyone done with it? Should I answer it, or anyone wants to boggle their mind? I'm giving a hint. I'm giving a hint that there are 4 lines in this FASTQ, and it is fixed. This is actually uh, you have to now think of. There are 4 lines in FASTQ.
Maybe we can give a command to exclude the symbols and exclamatory marks, so it only the sequences will be remain at the end.
If the sequences remain at the end, what happened? you will get all the lines with the sequences, and you have no identifier here to understand what how many reads are in the fast queue. Should we remove the quality line? If you remove the quality line, that can a bit tricky task, that you remove the quality line, then you find the at the rate only. That can work. It's not an issue, but it's again an issue how you are going to remove the quality line, by maybe using percentage, by maybe using star something which is not present here. You can remove it, but it's a long process. So the shortest way is oct nr percentage 4 equals to, yeah. So what is anyone
Can we count the
speaking?
total number of lines and divide by 4?
Yes, that is the Krishnaveni. Okay, you are actually correct. There you have to count the total number of lines in the fast queue. Those total number of lines have to be divided by 4, because the number of lines for fast queue are fixed. Header, sequence, separator, quality, these are fixed. So if you divide, yeah, everyone is answering now. So you have to divide it by 4. After dividing it by 4, you will come to know how many reads are actually there in the complete file. So now your answer is this. This is actually answer, counting number of lines and dividing by 4. This is the syntax which can be used. Though if anyone wants to understand how this syntax has been written, then I can explain, but you can do it later on also. You can copy it and you can use it, though it's really very simple. Then the second is, suppose you scan the file and find exactly 100 at the rate symbols. Does this mean the file has exactly 100 reads? Why or why not? That you guys have answered now. You are understanding it now. So you can answer again. Suppose you scan the file and find exactly 100 at the rate symbols. Does this mean the file has exactly 100 reads? No. Yeah. Very good. Dimple, Shivangi, Soumya, Krishnavi. Yeah, correct. So of course, it doesn't mean that there are 100 at the rate 100 reads. Now next question: how many reads are there in this image? If you see this file, how many reads are there in this line, in this image? Can you tell me? Yes. Correct. So what the actually, everyone is correct. There are 4 reads. This is amazing that you are understanding. The Samiksha, Shivangi, Suriksha, Pankaj, Ishita, Mitali, Bhakeshwar, everyone is answering correctly. Very nice. So are you anyone is left who is not able to understand? If is anyone left, so for that I just repeat. There is at the rate, there is a sequence, separator, and then there is again, this is a quality score. So this is a first rate, then again at the rate, sequence, plus, and this quality score. This is second, like third and fourth. So this is not that plus, after plus there cannot be anything. This is possible that after plus the same header can be written here also. So it's not like that always plus is not accompanied by anything. Now, should we move forward? Or you need a break? Each line in sets of 4. Yes, Kargi, you are correct. So, guys, do you need a break, lunch break, or should I move forward? Answer.
We need the break.
You need the break? So, okay, there are only 3 formats left. So, we will meet you after a break. So, I am giving you, okay, we'll